Hello, Dr. Martin again, and we're picking up with the second part of our lecture on Chapter 9. We've already finished 9.1. This is 9.2. And this kind of talks about some of the practical implications of meiosis, or the formation of sperm cells, egg cells, gametes, and how they are translated into the resulting zygote after fertilization. Um, for instance, it's kind of interesting to talk about twins. Um, we don't normally in science talk about fraternal or maternal twins. Instead, we talk of what are called dizygotic versus monozygotic twins. Dizygotic twins are twins that developed from two completely different fertilized egg cells. Two different egg cells, two different sperm cells. Dizygotic twins, di meaning two different zygotes. Really, dizygotic twins are no different than other siblings that were born at different times. It just happens to be siblings born at the same time. Two different zygotes, two different egg cells, two different sperm cells. Um, that's different from monozygotic twins. We would call those identical twins. Monozygotic twins, mono meaning one, monozygotic twins develop from the same fertilized egg cell, same sperm cell, same egg cell. We call these monozygotic twins. Now you could imagine that monozygotic twins have a lot more genetic information in common than dizygotic twins. In fact, we say it this way: monozygotic identical twins have 100% of their genetic material in common because they have the same egg cell, same sperm cell. Dizygotic twins, different sperm cells, different egg cells, so they have 50% of their genetic material in common, just like other siblings born at different times. So these are the kinds of things we want to talk about in the second half of chapter 9. Are some ways that genetics on the chromosomes of your cells translate into physical appearances. And monozygotic twins and dizygotic twins are some examples of that. Some great studies that have been done over the years are what we call twin studies involving monozygotic twins. It's great reading if you ever get some residual time in your daily life, your busy life, to do some interesting reading. You should look it up in Google or whatever. And what we have done in the United States and Canada is we've identified monozygotic twins that were separated at birth and grew up and matured in completely different environmental conditions. By environment, we don't just mean climate, we mean familial conditions. Um, Maybe never met in life. These are what we call twin studies. There are some very interesting medical issues that we can talk about. In other words, are some of our disorders or diseases, do they develop as a result of our environment or as a result of our genetics? You see where I'm going with this. If the same disease, disorder, condition, abnormality occurred in the same two twins that were raised in completely separate, different environments, then we would say that that would be genetically induced or genetically impacted. If a disorder or an abnormality, a cancer, a disease developed in a very low frequency in one twin versus another, that where they were raised in different environments, then we would say that maybe that disorder is more environmentally induced. So you understand the importance of twin studies and the interest of twin studies involving monozygotic twins. But anyway, we have to move on. There are some things that we have to talk about in meiosis here. There are obvious differences between mitosis and meiosis. I want to highlight them here because it's very important for you. You'll be tested on this. Okay, remember mitosis. A parent cell divides into two daughter cells, identical genetic information. The two daughter cells are clones of that parent cell. This occurs in growth, development, and repair of injury. Meiosis, on the other hand, results in sex cells. It occurs in sex organs, ovaries, females, testes of males. Here we have a parent cell, we divide it twice, and it ends up in haploid sex cells, gametes, pollen, spores, what we call germ cells. And those result potentially in another organism after fertilization. So you see the difference between the two. Mitosis occurs in our body cells, what we call autosomes, or body cells, somatic cells, whereas meiosis occurs in sex cells only. Sometimes errors occur. If errors occur during mitosis, they can result in mutations. Remember, we talked about that at the end of chapter 8. We talked about some environmental conditions that can cause nuclear damage, chromosome damage, UV light, ultraviolet rays, radiation, some drugs, some chemicals. So there are things that can cause genetic mutations. Some things can also cause mutations in sex cells. Mutations in mitosis tend to result in cancerous growths. Mistakes in meiosis tend to occur in what we call mutations. And one of the most classic ones is what we call non-disjunction. Remember the chromatids copied chromosomes, one from your mother, one from your father. They duplicate, but during anaphase, they fail to separate. And instead, the whole homologous chromosome will migrate towards one end of the cell. Cell divides. Now we have abnormal numbers of chromosomes. We call this aneuploidy, abnormal numbers of chromosomes, and it's caused by a non-disjunction, and it's where chromosomes fail to separate. In this case here, and this is meiosis 1, we have two twin homologous chromosomes. Normally they would separate during anaphase 1 of meiosis, but they don't. These two chromosomes, now there's other chromosomes here, we're just pointing out one. Here it doesn't, and so both of those twin chromosomes go to one resulting daughter cell. Then it happens again in the next meiosis. Okay, and so what we end up with, we end up with abnormal numbers of chromosomes. The zygote, the resulting zygote after fertilization, will have abnormal numbers of chromosomes. We call this aneuploidy, and this is as a result of what we call non-disjunction. It can occur in anaphase 2 as well, and resulting in abnormal numbers of chromosome. It can be lethal. In fact, if it occurs on some of the larger chromosomes, it tends to be lethal. The larger the chromosome, the more genes, the more information. But some of the smaller chromosomes, abnormal numbers of chromosomes, they tend to survive. And some of them are some of the more common genetic mutations in the United States anyway. So they can be lethal. In fact, normally they are lethal, but on some chromosomes they are not. Notice this karyotype. Remember what a karyotype is? It's a graph of a person's chromosomes. Normally we have 46 chromosomes, or 23 pairs. The first 22 pairs are autosomes. The last pair, sex chromosomes. Notice in this karyotype right here, this would be a normal karyotype, but sometimes individuals will have 3 chromosome number 21s. We call this trisomy, triminy 3, trisomy 21. It's commonly known as Down's syndrome. The latest numbers I've seen, it occurs about 1 in every 800 births in the United States. It's probably the highest genetic mutation in the United States. Not as many genes on this chromosome than this chromosome. See the difference? The size difference? And so on these smaller chromosomes, on non-disjunction, they can survive and lead lives that contribute to families. And you probably know a Downs individual, a trisomy 21 individual. They have very specific genetic characteristics. They tend to have some of the same characteristics. You know why? Because they're all on the same chromosome. So all of them have a third chromosome, number 21. There are some other conditions, Edwards syndrome, some other conditions, Patau's syndrome, chromosomes number 17, chromosomes number 13, but usually non-disjunction can be pretty lethal. But there are some cases in smaller chromosomes where non-disjunction does survive. The fetus will survive. Here, also some non-disjunction that occurs among sex cells, the X chromosome and the Y chromosome, where they simply fail to separate during anaphase. Instead, both sex chromosomes go to one of the daughter cells, and we end up with aneuploid conditions in sex chromosomes. Here's the triple X chromosome. We have what we call a Klinefelter's syndrome, where we have a male. If the Y chromosome is there, then it is a male, but they have an extra X chromosome. We call this Klinefelter's syndrome. About 1 in 750 male births. They tend to be viable, but they have some sexual underdevelopment, large hands and feet, some development of breast tissue. There's another one down here, Turner's syndrome. Only one X chromosome, and that's it. They don't have another sex chromosome. About 1 in 2,000 females, short stature, sexual underdevelopment, infertility, sterility, oftentimes. Look here, what we call Jacob's syndrome. We used to call this super male syndromes. It's a male, have X, they do have a Y chromosome, but they have an additional Y chromosome. About 1 in 1,000 males. Very few noticeable symptoms, maybe slightly taller stature, maybe persistent acne, but otherwise lead very normal lives. So anyway, you see, so there can be abnormal numbers of body chromosomes, some somatic chromosomes, but there can also be abnormal numbers of sex chromosomes, and we call these non-disjunction conditions. Errors can also occur on the chromosomes themselves, not just in chromosome numbers, but in chromosome material as well. In this case, we have a couple of chromosomes here with A, B, C, D, E, F, G genes, and then all of a sudden we end up with A, B, C, D, and we're missing something. And something may be added to another chromosome. Sometimes this may occur during what we call translocation or inversions or deletions. Here we've lost some genes. Here some genes have been duplicated onto another chromosome. So again, similar situations. This can occur on any of our chromosomes. It's not as common as some of the other the non-disjunction mutations, but it can be common. So here's the deal. These are, if mutations occur during mitosis, gamma rays, X-rays, UV light, chemicals, those can result in accelerating mitosis and accelerating the growth of cancers. But here in meiosis, we're resulting in human gametes. These are resulting in organisms after fertilization. So it's very important that we have pretty exact replication and the process occurs very smoothly and very normally. Here's another difference here that we want to talk to you about. This is what we call spermatogenesis. This is in males. It occurs in the testes of males. Notice we have two divisions. Okay, we have parent cell, doubled chromosomes. We split it, and then we're going to split this cell again. So what we end up with in males are 4 sperm cells of equal size, equal content, equal genomes. It's what we call spermatogenesis. In females, I want you to notice the difference here. We have, here's the parent cell, doubled chromosomes. We divide it twice, or we divide it once in here, and then we're going to divide this one again. And instead of ending up with 4 gametes of equal size, we end up with 3 small, what we call polar bodies, and 1 large egg cell. Do you remember on a couple of previous slides, we showed you a sperm cell attaching to an egg cell, and the huge discrepancy in size between a sperm cell and an egg cell? This is why. In meiosis of females, there's an unequal division during telophase. Chromosomes all separate just normal, just like in sperm cells, but there's an unequal volume difference of the cell. So what we end up with is 1 large egg cell and then 3 smaller, what we call polar bodies. They tend to not survive. They get absorbed into the uterine wall, but 1 large egg cell survives. That's a key difference between the development of sperm cells in males and the development of egg cells in females. Now, don't forget, we tend to humanize this, and we tend to make sperm cells and egg cells, but don't forget, meiosis also occurs in plants. It occurs in ferns, some of our primitive plants, and they don't necessarily produce sperm cells, but they'll produce spores. Fungi will do this. Plants will produce pollen. So it's all the same in meiosis, though. So we just tend to talk about humans more in meiosis than we do in other organisms, but still occurs in any organism that reproduces sexually, is meiosis. Hey, that concludes our lecture for Chapter 9. Hope you enjoy your week. Don't forget there's some labs to complete this week and also a quiz to complete this week over Chapter 9.